Now, apart from the sarcomere, the other important functional component of the skeletal muscle is the sarcotubular system. Now, this is in turn made up of two different components. One is the sarcoplasmic reticulum, which is modified to form long tubules called L tubules or longitudinal tubules. Now, as you can see in this diagram, these are long sleeve-like structures which enclose the myofibrils. So they surround the myofibrils. Now you can see that their ends are dilated to form the terminal cisterns or the lateral sacs, and calcium ions are stored in these lateral sacs. This calcium is required for contraction. The other component is the T-tubule or the transverse tubule. Now the T-tubule is nothing but a tubular invagination of the cell membrane, that is the sarcolemma. So the sarcolemma at regular intervals dips in transversely to form the T-tubules. And you can see that these are very closely associated, the L-tubules and the T-tubules. Now this is a more detailed diagram of the sarcotubular system, which is made up of the T-tubules and the L-tubules. So the sarcolemma dips in to form the T-tubules, and these are the L-tubules with the dilated ends that are called terminal cisterns, which are filled with calcium ions. Now there are two important receptors involved here. One is called the DHP receptor or the dihydropyridine receptor. This is present on the T-tubule. The other is called the ryanodine receptor and is present on the terminal cistern. Now both are calcium channels, but the ryanodine receptor is an intracellular calcium release channel and the DHP receptor is a voltage gated calcium channel. Now you can see that at rest there is no contact between the DHP receptor and the ryanodine receptor. So the T-tubules and L-tubules are not in contact with each other. But during muscle contraction, the foot plate of the DHP receptor moves towards the ryanodine receptor and makes contact with it, opening the calcium channels here, and therefore calcium leaks into the sarcoplasm. This calcium is made available to the actin and myosin for muscle contraction. So as we just saw, the receptors play a very important role in mediating the process of muscle contraction. So on the T-tubules, we have the DHP receptor, which has a rather unique foot plate, which can establish contact with the ryanodine receptor during muscle contraction. The L-tubules have the ryanodine receptor, which is an intracellular calcium release channel. Now, it's quite natural to assume that muscle contraction happens because of shortening of the actin and myosin filaments. Now, that is not the case. There is a rather unique theory called the sliding filament theory to explain the process of muscle contraction. So during muscle contraction, we know that myosin binds to actin. The cross bridge part binds to actin and then moves in an arc fashion. And therefore, the actin is made to slide over the myosin and it starts moving closer to the center of the sarcomere, bringing the Z lines closer. So there is no shortening of the actin or myosin filaments. Actin only slides over myosin. What does shorten is the sarcomere because the Z lines are pulled closer to the center. Now to explain that diagrammatically, during muscle contraction, the actin or the thin filaments do not shorten. The myosin also does not shorten. Actin only slides over myosin. So actin comes closer to the center of the sarcomere. It's sliding over myosin. In the process, the Z lines are pulled closer to the center and therefore the sarcomere shortens. Now that we have understood the functional components of the skeletal muscle, that is the sarcomere and the sarcotubular system, we can look at the process of excitation, contraction, coupling. What do we mean by excitation, contraction, coupling? Now this refers to the sequence of events by which the muscle action potential is converted to muscle contraction. So this muscle contraction at the molecular level is nothing but cross-bridge activity. So you can define excitation, contraction, coupling as the sequence of events by which the muscle action potential is converted to cross-bridge activity. So what is happening is an electrical signal is finally being translated into mechanical activity. Let's now study the sequence of events in excitation, contraction, coupling. Now we know that as a result of neuromuscular transmission, the nerve action potential has been translated into the muscle action potential. So the first step in excitation, contraction, coupling is the propagation of the muscle action potential along the sarcolemma and then into the T-tubules. Now this leads to activation of the DHP receptor. So when the muscle action potential reaches the DHP receptor, which is a voltage gated calcium channel, there is opening of these channels leading to entry of calcium ions here. Now these calcium ions cause movement of the footplate of the DHP receptor to the ryanodine receptor. So the DHP receptor now establishes contact with the ryanodine receptor, leading to a conformational change in the ryanodine receptor. So this in turn leads to the opening of calcium channels on the lateral sacs or terminal cisterns. So the calcium ions now move out into the sarcoplasm from these lateral sacs. This calcium binds to troponin C. This activated troponin C will in turn activate troponin T. And we know that troponin T is bound to tropomyosin. So it pulls away the tropomyosin from the actin, exposing the myosin binding sites on actin. Now that actin can bind to myosin, there is muscle contraction, which at the molecular level is referred to as cross-bridge activity. So what does this cross-bridge activity involve? Now at rest, we know that the myosin head has two binding sites, one for actin, which is free, and the other is to bind ATP. So ATP is already bound to the myosin head. Now the myosin head acts as an ATPase, so it breaks down the ATP into ADP and pi. Now this energy remains stored in the myosin head, so we call this as the energized myosin head, and this is represented by the star. Now once calcium is made available, actin can bind to this molecule. So we represent that as AM ADP pi with the stored energy. The energy is still not utilized, but actin is bound to myosin. Now the third step is the most important step, what is referred to as the power stroke. This involves an arc-like movement of the cross bridge, and therefore actin slides over myosin. So this requires energy, and the stored energy is now used up. So actin and myosin are still bound to each other, but ADP and pi are released. The energy is used for the movement, which is referred to as power stroke. Now actin must detach from myosin so that it can bind to a new site and continue to slide. In order to detach, a new molecule of ATP should bind to this complex. So once that happens, actin detaches from myosin and we're back to square one with myosin bound to ATP. So this cycle is repeated until the actin has sufficiently slided over the myosin to generate enough power for contraction. So this is a diagrammatic representation of the cross-bridge activity. So you can see that at rest, the myosin is not bound to the actin, but it is bound to ATP, which has already been broken down into ADP and pi, and the energy is stored in the myosin head. So this is the energized myosin head. Now, once calcium is made available, you can see that this cross bridge is now bound to the actin. Now, in the third step, you can see that the cross bridge has moved in an arc-like fashion, and therefore actin has slided over myosin. This is the power stroke. In this process, energy is utilized and the ADP and pi are released. Now the actin is still bound to myosin, so in the final step, a new molecule of ATP binds to the myosin head, and that is when actin detaches from the myosin head. So now we are back to square one. This cycle continues until sufficient power is attained. Now this is a diagram to show how troponin regulates the process of muscle contraction. So once calcium binds to troponin C, it activates troponin T, which in turn pulls off the tropomyosin and therefore removes the troponin I from the myosin binding site of actin, and therefore exposes this site for contraction. So you can see that in this diagram, this site is exposed because calcium is now bound to troponin C. So how is the cross-bridge activity terminated eventually? Now the moment the muscle action potential terminates, there is no further release of calcium from the L tubules into the sarcoplasm. So the concentration of calcium in the sarcoplasm falls, and this leads to a reversal of the events. So tropomyosin eventually slides back to cover the myosin binding sites on actin. Now the calcium which was released is pumped back into the lateral sacs. That takes a little longer, and this leads to relaxation of the muscle. Because it takes much longer for the calcium to be pumped back rather than for the release of calcium, the relaxation period is longer than the contraction period. So this is a representation of the cross-bridge activity which we have already seen. Now this is a summary of the entire excitation contraction coupling process. So the nerve action potential is first converted into a muscle action potential via neuromuscular transmission. And now the process of excitation contraction coupling begins. So the muscle action potential travels along the sarcolemma, reaches the T tubules and activates the DHP receptor. The footplate of the DHP receptor binds to the ryanodine receptor, and this opens the calcium channels in the terminal cisterns, leading to an efflux of calcium ions into the sarcoplasm. Now the calcium is made available to troponin C, which will in turn activate troponin T, and the tropomyosin is pulled off from the myosin binding site on actin, and thus the cross-bridge activity is initiated. Let's now look at how smooth muscle organization and contraction differ from that of skeletal muscle. Now we know that structurally smooth muscle cells are spindle-shaped and have single nuclei, unlike skeletal muscle cells. Also, there is no motor end plate, and each axon is surrounded by many smooth muscle cells. Now, though both thin and thick filaments are present in smooth muscles as well, these are not organized into sarcomeres. So there are no Z lines, and therefore the actin filaments are anchored to what are called as dense bodies present in the cytoplasm. The actin filaments are arranged diagonally, and there is no organization into sarcomeres. Also, if you look at the actin-myosin ratio in smooth muscle, it is quite high as 10:1 when compared to 2:1 in skeletal muscle. So this means that the myosin content is low. Now, smooth muscle contraction considerably differs from skeletal muscle contraction. First of all, the source of calcium for contraction is not merely the sarcoplasmic reticulum, but also the extracellular fluid. Also, the sarcoplasmic reticulum in smooth muscle is not organized into L tubules. The second important difference is that there is no troponin in the smooth muscle. Instead, the regulatory protein is a molecule called calmodulin. So calcium binds to calmodulin, and this calcium calmodulin complex activates an enzyme called myosin light chain kinase, which causes phosphorylation of the myosin head, and now the myosin binds to the actin, leading to contraction. Now, the ATPase activity is also very slow, which means that the contraction velocity is low, and therefore smooth muscle does not undergo fatigue easily. Finally, the relaxation occurs by dephosphorylation of the myosin head, which is brought about by an enzyme called myosin light chain phosphatase. Now, this enzyme, myosin light chain phosphatase, can also dephosphorylate the light chains while myosin is still attached to the actin filaments. So this produces an alternate cross-bridge cycle which has slower kinetics, and this is what we call as the latch state. Now, the importance of the latch state is that the muscle can remain contracted for long duration of time with little expenditure of energy, and that is very important for the functioning of smooth muscle, example in the GIT or in the urinary tract and so on. Now, this is a diagrammatic representation of the process of smooth muscle contraction. So the source of calcium is both the extracellular fluid and the sarcoplasmic reticulum, which is not organized into L tubules. Now, once calcium is made available, it binds to calmodulin and forms the calcium calmodulin complex. This activates the enzyme myosin light chain kinase, which in turn phosphorylates the myosin head. So now the myosin head can bind to actin and cause muscle contraction. Now, the process of smooth muscle depolarization is also different from that of skeletal muscle in some ways. For example, depolarization here is brought about by entry of calcium ions and not sodium ions. Secondly, both the graded potential and action potential can lead to calcium entry and therefore depolarization and contraction, unlike the skeletal muscle where only an action potential can do this. And finally, if you take single unit smooth muscle, it is capable of generating pacemaker potential, which is not seen in skeletal muscle at all. So with that, we come to the end of excitation contraction coupling. Thank you.